Welcome, everybody, to another in this series of reflections on the history of modeling and theory. James O'Dwyer and I are here today. We are lucky to have Warren Ewens with us today. And before we start, and I'm going to launch into a brief biography of Warren, I just wanted to shout out to Kaylee Charlebois, who's been working behind the scenes and really helping us turn this podcast into a YouTube video. So thank you, Kaylee. Dr. Warren Ewens received his PhD with Patrick Moran of the Moran model stochastic process from the Australian National University in 1963. And he has held professorships at the University of Pennsylvania, La Trobe, and Monash universities, kind of going back and forth between Australia and the United States and shaping both fields. He's named the Christopher Brown Distinguished Professor of Biology at the University of Pennsylvania, where he spent much of his career. Warren is an officer of the Order of Australia and Fellow of the Royal Society of London. He's received a number of other prizes, and I'll just highlight two: the Australian Statistical Society's Pittman Medal and Oxford University's Weldon Memorial Prize for noteworthy contributions in the development of mathematical and statistical methods in biology. I wanted to show and tell. This is my copy of Warren's 1979 Mathematical Population Genetics, and it is dog-eared. And it is I have written and examined it over and over again over the years, and really learned about kind of the core mathematical and statistical foundations of population genetics from this book. I read it as a graduate student, and I turned to it again and again and again over time. So it's really shaped my understanding of the field. And so thank you, Warren, for writing this book. There's a 2004 update to it, as well as a second volume on more data-driven theory. I'll just highlight a couple of classic papers that have had a huge influence on the field. His 1972 paper in TBB on the sampling theory of selectively neutral alleles provided the statistical null distribution for the number of copies in a sample of each allele to be expected under neutrality. This is now called the Ewens sampling formula after Warren.
And Sally, I don't want to interrupt you there, but something probably where I first came across Warren Ewens' name was this kind of work, but not in its original setting, but instead applied in ecological communities. And so I saw that Ewens sampling formula crop up maybe somewhere he never expected it to be used.
Right, interesting. Exactly. He also has developed a lot of the really important tools in human genetics to be able to associate genes with particular diseases and disorders. And that's been a problematic thing because correlation doesn't imply causation. And two things can be correlated just because of population structure over space. They just so happen to have risen over here together. And that can mislead in association studies. So he developed this, what's called the transmission disequilibrium test, that looks at transmission from parents to offspring, as well as linkage disequilibrium. So he can tease apart what is happening due to structure and what is happening due to linkage of closely linked sites. And so that his 1993 paper introducing this to the field has been cited over 5,000 times and was able to demonstrate with stronger statistical power that insulin-dependent diabetes is associated with the insulin gene on chromosome 11. He's worked on everything from clarifying the meaning and limitations of the fundamental theorem of natural selection to accounting for population size change in our stochastic models of drift and establishment probability for beneficial alleles. Again, in my opinion, Dr. Warren Ewens has shaped modern population genetics and the mathematical structure of it, and I'm really looking forward to chatting with him today.
Great.
Welcome,
Hi, Warren.
Warren. Hi.
Hello, how are you, Sally, and how are you, James?
Great. I'm so pleased that you could join us today.
Yeah, we're doing great here. Thank you for joining us.
Okay.
I'll ask the first question. Warren, you're coming from originally quite a mathematical background. So we're wondering, what stage was it that you realized math was a tool that could be used to provide insights in biology?
Yes, okay. So, James, as you said, let me start by pointing out that I had a very unusual background so far as genetics and biology is concerned. I never, even once, at any time in high school, took a course in biology. Up to and including my PhD work, I was essentially totally focused on math and stat. And that's in the British system, which we had in Australia, for good or for bad. You focused early what you could do, and what was your strength, and you sort of left aside a lot of other things, which goes against the the American system of breadth and all that sort of stuff. So it was slightly amazing when in 1972 I was appointed as a full professor of biology at Penn, never having taken even 1 minute of a biology course any time in my life, high school or university. So anyway, what I did in my PhD was to consider some purely mathematical and statistical problems in population genetics without thinking about their relevance to actual biological situations, which I wouldn't have known much about anyway. And that's not really an unusual situation. Problems are often given to PhD students which are to some extent artificial. What a PhD student is often asked to do is to show, or perhaps not show, her ability to answer specific, well-posed questions without necessarily thinking about the potential relevance of the answers to those questions and of the relevance of those questions themselves. And now to get to the point, James, thus I had no way of judging the relevance of what I learned in math and stat, no idea of the relevance of that to biology until well after I finished my PhD. My PhD thesis and work had been totally focused on those artificial math and stat type questions. Now, I want to say a bit more on this to answer your question about my post-PhD activities. Having finished my PhD, I realized the narrowness of, I realized I want to get into population genetics, and I realized therefore the narrowness of the background which I had had in my PhD training, as I said, totally focused on essentially math and stat. So I made a point of reading all the relevant literature, mathematical and theoretical and sort of practical, that I could find. I took about 2 years doing almost nothing except that. And it was when I read that literature, and now I get around to answering your question, that I realized the power of, for example, matrix theory, in particular as applied to Markov chains, diffusion theory, backward and forward equations, for example, to solving problems on the theoretical aspects of evolutionary genetics. And in particular, I read the stochastic, I was interested much more in the stochastic rather than the deterministic theory for a long time. So in particular, I read the stochastic theory of Fisher and Wright, and I noticed that not even did they never use the expression Markov chain, but then they seem to be totally unaware of Markov chain theory or matrices as a whole. And therefore, they made some errors in their computations. And this is also true of some of their work in diffusion theory, which was sometimes wrong. And these errors were later pointed out to Kimura, whose work I read very thoroughly. Kimura pointed out these errors. So in reading Fisher's work on the correlation between relatives, not really an evolutionary matter, but it's it's in population genetics, but also in his work on evolutionary genetics, I became aware of the great importance of the concept of the additive genetic variance, which I would call a sort of a purely mathematical concept. Hang on, let me just get onto my notes here. A purely mathematical concept, but nevertheless central for many areas of genetics and applied genetics. So I would say, in answer to your question, that when I learned all about the additive genetic variance and its broad relevance to population genetics, and when I understood the power of matrix theory and diffusion process theory, that was when I first realized the power of math and stat to address real practical population genetics problems.
Great. Just to follow up on it briefly, Warren, it sounds like you reached a point where you knew there was something missing, but it then took that, couple of years of reading the literature. It sounds like there was something you felt that was missing with your purely theoretical PhD work, and then it took that time to find out.
I realized it was missing. And I also realized, and I'm going to come back to this a bit in a minute, it's part of recognizing your strengths. I also realized I wasn't really up there with the top level mathematical and statistical people, and that I could be better involved doing things in a different way, which I'll get onto in a minute. But I did realize that narrowness, and that's why I spent a couple of years after finishing my PhD in the broad reading, which I just mentioned.
I think that also shows a certain humility. We can't possibly know everything, and when you are bridging across fields, you have to be humble and take the time to learn. So
yes, that's a point I'm going to be emphasizing quite a bit in a few minutes.
Yeah. So I mentioned in the introduction that you worked with Moran. I'm curious about that, but I want more generally to ask you, who were the people, either personally or through their research, that inspired you in your career?
Yeah, well, I'm going to say quite a bit about this question. So, by the way, you said Pat Moran. That's how you pronounce it.
Oh, I didn't know that. Moran process.
Pat Moran. And of course, Pat Moran was my PhD supervisor, so he was an influence. But he wasn't really a direct influence on me in my PhD. His idea of supervision was to just ignore me. You sink or swim. It's a sort of the Cambridge model. If you can't do it on your own, buddy, you can't do it, okay? So most of the problems I worked on in my PhD, he didn't even suggest to me. I thought, this is an interesting mathematical problem. Let me do that. For example, one thing I spent a bit of time on my PhD is, many Fisher-Wright model results, matrix theory results. You cannot solve many things, but you can get extremely good approximations with diffusion theory. And so one of the problems I set myself was, how accurate are those diffusion theory results to the matrix theory results? So of course, Pat was an influence, you might say, but his main influence was the following. Your work has to be at a world level. We don't want to produce people who are parochial and focusing on just what you might call narrow Australian issues, like thinking about koala bears or platypus. You have to think about things which relate to the whole world. He said, he was extremely smart, and he said, I'm not here to train people unless I'm training them at a world level. And that was a real, I think, challenge to me and to all his graduate students. That's what he aimed at. Okay, so now other influences, and I'll go through them. And as I said, I'm going to talk quite a bit on this particular question because it gets to some of the other things which you have on your list of questions. So secondly, Sam Carlin.
Yes.
Did a postdoc with him in the first 6 months of 1964 at Stanford. He taught me some pretty high-level stochastic process theory, for example, multi-type branching processes. He was very, very friendly to me. We became close friends for 50 years, unbelievably, just like that. And in fact, we remained close friends for the rest of his life. He helped me a lot how to approach doing research. I'm eternally indebted to him. Although I do recognize some of his, I would say, limitations in some way. Secondly, Jim Crow. I met him on January the 3rd, 1964, 2 days after I arrived at Stanford to do that postdoc. Crow was giving a seminar on January the 3rd at Stanford. So within 2 days of arriving in America, I met Carlin, McGregor, Crow, Bodmer, and Cavalli-Sforza, and quite soon after that, Kempthorne, who I'll say a lot about soon. So I really got thrown in at the deep end, which actually was very good for me. So I admired Crowe very much from the point of view of his very broad thinking about population genetics, both practical and theoretical. And he was very generous to me in discussing those matters, and he influenced me a lot. He invited me to stay in Madison on 2 occasions for substantial amounts of time when we did that. However, later on, I often disagreed with a lot of his views, for example, on genetic load and other matters, which are not perhaps worthwhile talking about now. And it was a real agony for me to say in print, that I disagreed with this, that, or the other thing which he said. I felt very sad about that. Third, I have to mention Moto Kimura. His many high-level and very influential papers in the 1950s and the 1960s, I read as soon as I could. But I had very little personal contact with him. I didn't meet him until 1973. And some of our contacts were antagonistic. We would often fight about something. I have to tell you now, I won all those fights, although he would never admit that. And now, last but not least, Oscar Kempthorne. Now, as I said, I met him at Stanford in 1964 when I was doing that postdoc with Sam Carlin, and he was on a sabbatical leave, I think with Carlin. Kempthorne was very derisive of the Stanford school. So you better watch out, Sally, because you're going to be indirectly under attack here.
Okay.
He was very derisive of the Stanford school and what he called the ivory tower viewpoint, which they had, just up in the math. He was thus sometimes derisive of Sam Carlin, privately to me, but never in public. I became very close to Kempthorne. So being based at Iowa State, Kempthorne was involved, amongst other things, with things like plant and animal breeding programs, quote unquote. Your work should have some practical relevance of this kind. Get your hands dirty. That was what he would say to me. Okay. And I'm going to mention, as I told you, I'm going to talk a lot about this because I'm going to talk about 3 things which arose from Kempthorne's pretty blunt comments. First, I decided, since I finished my PhD, that apart from learning more about mathematical theory, as I mentioned earlier, I would also spend, and I mentioned this before, 2 years or so reading the population genetics literature generally, especially those aspects of the theory which had a real-world application, not just the mathematics, but the biological application. So amongst other things, I read about Fisher's concept of the evolution of dominance, about self-sterility alleles, about the effective population size concept, I'll come back to that a bit later, about the genetic load concept, which I mentioned before, and about the fundamental theorem of natural selection, which I again will mention in a few moments. I found that much of the mathematics discussing those practical issues was wrong. And in these areas, and some of my earlier work was involved in correcting it. I'll say more about that in a minute. That, of course, leads to the accusation that people could say is, well, all he did was nothing new himself. He just did minor corrections on other people. But I was doing that as well as doing my own work anyway. So secondly, I actually left the evolutionary population genetics field in the early 1970s. And I moved to the application of statistics in human genetics, especially in linkage analysis and human disease genetics. And I did this because my friend in the medical school, Richard Spielman, said much the same thing as Kempthorne said to me. Get out of that ivory tower and do something useful in your life for a change. Okay, pretty blunt words. That's what your friends say to you. So we worked together in that area for almost 20 years. And in fact, my most cited paper, almost twice as frequently cited as my paper on the sampling theory of neutral alleles, was on our creation of the transmission disequilibrium test, which was used and is still used very much in that area. So you could say that was, in a sense, part of Kempthorne's influence, thinking that way. So I found that and now I come to play to your strengths. I found that my most useful role was being a sort of a middleman between those in the lab, like Rich, and those in the field who I could talk to and understand, and between them and strong mathematicians like Carlin, and I'll mention others in a minute. I could talk to both of these people who perhaps couldn't talk to each other, so I could act as a useful middleman. And I'll say more about that middleman thing in a moment. So I'm answering your question, but perhaps somewhat indirectly. In 1994, I was in effect asked by some people at Penn to start a genomics program. And I spent a huge amount of time doing that. And at one time, I was chair of their genomics and computational biology graduate group. And here again, I found that I had a middleman role to play between lab geneticists on the one hand and computer scientists on the other hand, who couldn't talk with each other, but I could talk to each of them and therefore act constructively as a middleman. However, I did keep in touch, even though I was out of the field in the late 1970s and 1980s with population genetics. And here I also became a middleman during those years without really being very much involved in it. I met John Kingman in 1974, and I introduced him to some population genetics problems. He was by far the most powerful mathematician that I've ever been involved with, much more powerful than Carlin, for example, which is a pretty high level of power. However, he did make a few mistakes in applying that power to population genetics, as Jeff Watterson and I noticed. We had no difficulty in bringing that to his attention, and he accepted what we said very gratefully, in fact, positively. And this led to another middleman role, together with Jeff, forging a link with Kingman in his mathematical power and real-world problems. And to some extent, it led to his concept of the coalescent. So this was another middleman role, which I found was the sort of role that I liked best and where I could make my best contribution. So all of this eventually derived from Oscar Kempthorne. You asked who influenced me. Oscar Kempthorne and his pretty blunt views. Maybe you could say he was the most influential and the most inspiring person that I had as it related to my career path. Okay, so that's it on people who influenced me and what that influence meant.
Thank you so much, Warren. And I wanted to show my dog-eared copy of your book. And it's relevant to this because you also have been a middleman between the theory that was developed in the 1900s and students current and future. So thank you.
Well, I hope so. That's partly, of course, what I intended.
Yes.
So I'll move on to our next question. And I'm wondering now if this is going to be an example of one of those cases where you found something that was wrong or something that was your own work completely separate from that. So we're wondering if you have a favorite memory of an aha. Sorry, go ahead, Warren.
Yeah. Well,
yeah. So we just
so
wondered if
you're
you have a
asking me
favorite
to
memory
be
of an aha moment.
very sort of self-centered and so on. So my biggest aha moment was when I was working on the sampling theory of selectively neutral alleles. And I realized immediately I did that within 3 seconds of getting all the formulas, I realized that the procedures which had been used up to then to estimate the basic parameter theta were based on precisely the wrong part of the data. And as I said, I worked this out. The mathematics showed it to you. And I worked this out within 3 seconds. And that was a completely amazing aha moment. It took, I was working with Crow at the time. We were both together, as it happened by chance, in Texas. And I told him that. And of course, he was a bit, I don't know, upset by this, because he himself had been the main person working in estimating theta by the wrong part of the data, namely the gene frequencies rather than just the number, allele frequencies, rather than just the number of alleles, which is a sufficient statistic for theta and thus has optimality estimation properties.
Ah, fascinating.
That was an incredible aha moment. And I say it came just immediately. There was no, I mean, it wasn't very hard to find it, but it was still very aha.
So things like heterozygosity or expected heterozygosity, that you just immediately could see that wouldn't do it.
Yeah, that's right.
That's great.
And then other people, I had a very close friend called Harry Guess, who unfortunately died about 20 years ago. He was a PhD student with Jim McGregor, and I went and talked about this in Stanford in 1971. Wait there, let me get this right. 1971, that's right. After I'd got all the theory worked out. By the way, I had to get Sam Carlin and Jim McGregor to help with put the last nails in the coffin on that theory, because it got to a level beyond my ability, but it all worked out together. We talked it all through, and they did the really hard work. Anyway, Harry Guess was a student of Jim McGregor's. He came to my talk, and he did two things. I was working back in Madison with Jim Crow after that, and Harry Guess came by, and he did two things. Firstly, he gave me the Carlin and McGregor paper, which nailed home the final problems that I had with the sampling formula. And secondly, he'd done simulations, which showed that the standard deviation of the accuracy using the number of alleles was a good deal less than the standard deviation using the allele frequency. So that confirmed the theory. So Harry became a very good friend, and we wrote up a paper together on that in the following year or two.
Yeah, it's not intuitive, so it's nice when the math kind of shows it instantly to you.
It's one of the few occasions where I think the math knows more than you do, or something like that. I cannot listen. I know it sounds very self-centered. I just cannot even tell you how much of the whole thing blew my mind.
Right. That's great. Maybe on the other side, do you have a favorite paper of yours that you wish people, that one, so the Ewens sampling formula is really well known.
Well known, yeah.
Yeah, as James mentioned, it's used also in ecology. In the introduction, he mentioned that. But do you have something on the other end that you think is a valuable gold nugget in your work, that a paper that you wish were better known?
Yeah, I'll come to that in a minute. That's actually your next question. But let me say a little bit about why I left population genetics pretty soon after I developed the sampling theory formula. I realized it was not really all that useful, because it made, for several reasons, it made various assumptions like fixed population size, stationary behavior, mutation rates, of totally symmetric mutation rates, which I thought were pretty unrealistic to the real world. But also the whole purpose of it was to test the neutral theory, and I realized immediately it had very, very weak power. So I became a bit disillusioned actually following that formula, and that was why I was very happy to move with Rich Spielman into the human genetics area. But now to come to the question you just mentioned. Yes, I think there is one paper which I wish was better known, and this was my 1992 paper on optimality principles, an optimality principle in population genetics. I think it's got quite interesting results in it, and I actually would like to think through even more now what those results might mean. But it's one of the, I think it's one of the very few papers which really addresses optimality questions in population genetics in a correct way. There's a lot of trash in the population genetics literature on optimality. So I'm a bit disappointed, I have to tell you, but in answer to your question, a bit disappointed that that paper is not so perhaps well known, as well cited as I, from my very selfish point of view, would have wanted.
Well, we'll give it a bit of a lift, and we'll put a link into our notes for this video. So hopefully more people will see it.
Yeah.
Warren, do you think there's any reason why that was the case, or do you have any sense of maybe why that didn't become better known?
I don't know whether it's the answer to this your question, James, but perhaps part of the answer is it's a mistake to do something which is sort of too much high level or too much out of the norm. People are just going to ignore it. They're going to say, why should I bother about that? You've got to pitch the level of what you do at what people will want and what people will read. And maybe it missed out on those criteria.
Interesting.
Yeah, I mean,
Okay, I'm going to move on to our next question. We're wondering if you could wave a magic wand so that everyone better understood an existing result from evolutionary theory, which result would that be?
okay, now, James, I get to answer this question in a very, very selfish and self-centered way. And
That's okay. That's allowed. That's okay.
refer to my own papers, and the reason for that is they're the ones I know something about. So some of the papers that I wrote after my PhD, which I sort of half talked about, discussed matters which I think were better understood in population genetics theory. And these are, as I said, these are the ones which I think were perhaps better understood. And I'll give you two examples. First, there was and still is much confusion in the literature about the effective population size. So despite my trying to correct this, this confusion remains. One aspect of this, and this is something I'm going to send you in a few minutes. I told you in an email about it, Sally.
Mm-hmm.
It's 1931 initial concept of the effective population size. Nobody seems to realize this is what I called an eigenvalue population size. And practically nobody ever talks about that concept, but that's what Wright was talking about. That's very clear. Once you go through his work, that's the case. And so the eigenvalue population, this might be one which I wish was better understood. The eigenvalue population size concept, because it was what Wright was originally talking about, is to my mind not sufficiently thought about. One exception to this is your colleague Michael Whitlock, Sally, and his 1997 paper with Nick Barton, which had all this well understood. And second, I wish there'd been more discussion in the literature about Fisher's evolution of dominance concept.
Mm.
Work I did in that was totally ignored, though I showed that Fisher's calculations were totally wrong and totally misleading. But my paper was totally ignored. But I think that's probably because the whole evolution of dominance issue is a non-issue in the sense that people are not interested in that very much more. So, you know, I'm not surprised by that. So by contrast, now the other way around, I think that my work on the genetic load concept, which I thought was very badly thought out by many people, in particular the people who introduced it, Haldane, Kimura, and Crow, and which I therefore criticized, and my work on the fundamental theorem of natural selection, indicating exactly where Fisher made an error on that, have been as much recognized as I would have wanted.
Mm-hmm. And you're still working on that. One of your recent papers was on the
Yes.
fundamental.
That was a really fun paper to write. And there are a lot of, I often write papers in such a way that they're directed towards one person and telling the person, shut up, you're talking nonsense. I won't tell you who the person was for that paper, but I'll whisper it in your ear sometime or other when I next see you. It wasn't you and it wasn't James.
Yeah, no, I have so many questions about that, but maybe we can get to that later if there's time.
Well, we can get back to that perhaps when we've got through the regular list of questions.
That sounds good. For students entering, so many graduate students watch these videos, and for them, do you have any advice about how to approach theory and the insights it provides?
It's essentially what I said before. Recognize your own strengths and play to them, right? We all have different strengths and we all have different weaknesses, okay? And you play to your strength and use it. You have to remember also that many people have strengths greater than yours in this or that other area. And in particular, I hope there are not too many mathematicians listening to this. Many mathematicians should realize that there are many people just as smart as they are, let's say in the biological field, contrary to perhaps the typical mathematical point of view, that they're the smartest of the lot. You have to realize there are smart people in all fields. Mm-hmm. And you want to connect with them and use what you can, if you can be a middleman in getting involved with people on both sides of your own capabilities. That's my main and perhaps only advice.
Yeah. Do you think there's something about being a modeler and applying math to biology, to genomics, genetics that allows you to play that bridge?
I think that's a very perceptive comment, Sally, and I think the answer is yes. And developing your own model, trying to work them through, and on the one hand, seeing how far you can take them so far as the mathematics is relevant in the mathematical direction, and on the other hand, how far you can take them so they have some sort of biological relevance is what you should be doing. But doing the modeling yourself, as you said, I think is a very good starting point.
Thank you. I'm going to take the next question and then we'll go to a couple from James. So Warren, you know, I hope you know how much respect we hold for you because you help place population genetics and genomics on a much more rigorous mathematical foundation and shared that with us through your work and your books. So one of the things that you wrote recently was that the early population genetics was single locus theory, and that had to be bridged to a multi-locus genomics perspective to understand how evolution really works, which is not gene by gene.
Yeah.
I think that was a really insightful transition that you helped the field make from one gene at a time beanbag genetics to evolutionary genomics.
Yeah. Well, let me comment on a question you were going to pose earlier about what was it? Let me just see here. How do you think the perception of modelers and theoreticians of biology has changed?
Yeah.
I think it's much more realistic, less ivory tower than we were in the 1960s and 1970s. And I think that's because realism is forced on you by having to deal with data, and in particular the genomic data, which is, of course, swamping us all right now. The main work, I would say, in population genetics, I've made this statement many times, is retrospective, going backwards in time, using this data and ask what happened in the past to lead to these data, rather than what population genetics was in the 1940s, 50s, what might happen in the future. Look backwards. That's what I think is the modern modelers are doing and should be doing. But now to come to the two locus theory. And you asked, did I myself identify new areas? And I think the answer is no. I did not identify new areas in the, let's say, the 1960s and 1970s. And therefore, while I did a lot of work in two locus theory, I simply was following the trend and didn't identify myself as that important new area. So I became interested in the effects of linkage, recombination, and epistasis, and things of that nature, which you can only start addressing at the two locus level. Kimura led the field in all of that. His work was completely and totally outstanding. I've publicly ranked him with Fisher as, in my view, the leading all-time ever population geneticist.
Mm-hmm.
Ranking with Fisher is pretty high.
It is.
Now, two locus, this comes to what you said a moment ago, two locus theory was a natural evolution from one locus work. And more recently, I've been trying to go even further and develop aspects of population genetics theory at the whole genome level. And particularly to ask what sort of well-known one locus results, for example, like the Hardy-Weinberg law, actually continue to hold when they're viewed from the perspective of a whole genome evolutionary process. It's very, very difficult. I don't think I'll ever be able to get any results in this area, but just to keep my brain ticking over, I'm still trying to think about those things. So that's where I'm on that. I've really made essentially no real progress, but I'm still working on it.
That's great to hear. I'm sure we will. I'm sure you will make progress. I'm very confident. James, over to you.
Yeah, I have a couple of questions here, and so I phrased this next question as a revolution in terms of types of statistical prediction or methods. So I don't know whether you'll agree there is a revolution or has been, but I'm thinking here of people using machine learning or AI applied in biology And so I'm thinking about, well, I'm wondering what your perspective is, thinking about the different kinds of methods people have used. Do you think theoreticians of the future are going to be using very different methods than you have developed or that you have worked with?
James, I don't have a good judgment on that because I don't even understand AI or machine learning. I'm trying to understand it, but every time I ask somebody what is AI, I get a different answer from different people. So I still don't quite understand. I think you should open your mind to two possibilities. First, that stuff will revolutionize things and you have to learn to use them. You have to also open up your mind to the possibility that it won't get you anywhere. It's not going to be really useful in real practical problems where you've got to take into account all sorts of complexities which AI and other things, machine learning, might not be able to handle. I have no idea about the answer to that question.
That's why we ask it, because we don't know either.
We don't know either, but those two possibilities seem like the right ones to keep in mind, I think. Okay, and I'm going to take our last regular question as well. I mentioned in the intro that I had come across your work as an ecologist, reading papers on neutral theory applied in ecology. And so your work was foundational and very much used in that area.
Well, let me talk about
considering species rather than alleles. So I guess I'm wondering your perspective on the interaction of ecology and evolutionary theory.
I have three or four comments to make in that direction, James. First, Steve Hubble contacted me after my paper on the sampling theory, and he asked me whether I thought that my results would apply in ecology. He asked me whether I thought they would. Well, now, he was at Princeton at the time, and I was going up to Princeton fairly regularly because I was part-time supervising a PhD Princeton student, Karen Kirby. So I agreed to meet him next time I went to Princeton and discuss that issue. So we did meet and we did discuss. And what I said to him was that I did not think that the sampling theory, the stuff which I happen to have done in genetics, would be useful in biology. And I gave him two reasons for that viewpoint. The first, as I said a moment ago, is that that sampling formula relies on many assumptions like stationarity, fixed sample sizes, and so on, which were already unlikely to happen in genetics, but even more unlikely to happen in ecology, which is a much more complicated area. There's many more issues to talk about. So I just didn't think it would be practically useful. Second, even apart from that, my sampling formula basically derives, if you think through the logic of where it came from evolutionary principles. And you can see that best by seeing that it's best derived from Kingman's coalescent process. So I thought it's an evolutionarily derived idea. I wasn't too sure that there was a parallel evolutionarily derived thinking in ecology. And that was why I said to Steve, look, I just don't think what I've done has any use to you whatsoever. Despite that, he just went ahead and did it anyway. So much for my influence. Now, two other things, though. First, I'm aware of comments such as made by Crow and Dennis in 1988. There has been an increased interest in the effective population size because of its use in the study of rare and endangered species.
Right.
And so the idea there is that if a population loses genetic variation, which is what the effective what Wright's effective thinking in the effective population size was all about. If the population size loses all genetic variation and then perhaps becomes more vulnerable and perhaps lead to extinction, that has an ecological aspect. That species no longer exists. So there is a connection, or at least Crow and Denniston saw this connection between the effective population concept and the problems with rare and endangered species. So I think that could be followed up. I never followed that up. I knew that idea, of course, 40 years ago. I never followed it up. because you've got to choose what you're going to do. And I just didn't think that was my bag to do that.
Mm-hmm.
But maybe other people might be better placed than I am to follow that idea up. The other area where there's this interaction between genetics and ecology, I once had an interest in the concept of the so-called minimal viable population size. And I suppose that's an ecological concept, although frankly, what I read on it, I thought that many people didn't quite know what they were talking about anyway. Anyway, I suppose it's an ecological concept, and the preservation of genetic variation does come into the discussion on that concept. So again, there might be a link with ecology, which could be followed up between genetics and ecology. But again, I never followed that up. You know, what wasn't my strength. I had plenty of other things to do, but other people might have a good ability at pursuing those links.
Great. Thank you for those thoughts.
Yeah. Do you mind if I follow up on just a follow-up question on the fundamental theorem?
Yes, you certainly may.
So your 1989 paper really shaped my view on the fundamental theorem of natural selection by Fisher, which was it's not the fundamental theorem of evolution. It's just the fundamental theorem of natural selection. And it's even more limited than that by just saying if all else stayed the same, including the fitness of individuals is changing as gene frequency changes, that's ignored. But it is saying, how does fitness rise through the allele frequency changes if all else stayed equal? And I like that in a nutshell.
Sally, let me remind you of the first few words in Fisher's 1930/1958 book. Evolution is not natural selection. I forget exactly what it is. You know, he realized that there was much more to evolution, for example, mutation rates and so on. And you can see from the title of his book, he was going to focus on the effects of natural selection, the genetical theory of natural selection. And he realized from the first that that was a perhaps narrow thing to talk about, but that's okay. You're allowed to choose the field you want to talk about. I mean, you can talk about Chinese pottery and ignore Japanese pottery if you want to, you know, you're perfectly entitled to do it.
Right.
And I think that's what he did. Of course, he did talk about mutation, so he didn't ignore it, but his focus, I think, was on the effects of natural selection. So going on from that, I sort of kept on changing my view, and what's in my even 2004 book is different from what I now think. And that is, well, I think, and in fact, I think I proved conclusively in a paper I wrote last year that Fisher simply made a basic mathematical error. And just did an incorrect differentiation procedure. But let me say more about that. If you read, I spent God knows how many hours reading his 1958 book, and you talked about shop soiled books. You should see how shop soiled my copy is of that book. It's all torn and ripped. But what he did do, at least, he did not do this in 1930. But in the 1930 edition, but what he did do correctly in 1958 was a fully multi-locus analysis. It was also in continuous time. So if you wish to talk about that theorem, you must talk about a theorem on a whole genome level and in continuous time. And one of the people that I was sort of aiming the paper at were people who talked about it purely on a single locus basis and or purely on a discrete time basis, which I found much more easy to work with. So unless you realize that's what it was true. He did a truly multi-locus analysis. It was incredibly far ahead of its time. In 1958, he just made a very obvious mathematical error. So unless that's my attitude right now. It was a brilliant failure.
That's interesting because there's kind of two ways of seeing that you've voiced in the past, which is that it was a partial picture, but not the whole picture.
Well, let me tell you why I said that.
That it's an error, yeah.
Because I introduced the so-called partial change in fitness, And I proved something about that partial change. And I made a huge mistake, which I mentioned in my 2024 paper. In order to preserve Fisher's reputation, because I really thought incredibly highly of him. I met him, I'll tell you more about that in a minute. I'll tell you more about my interactions with Fisher and Wright, both if you want to. I wanted to say that's what he thought. That what I said, this partial thing was correct. And to save Fisher's reputation, I said in my 2004 book, that's actually what Fisher thought. That was a huge mistake because he did not think that very explicitly. I should never have tried that sort of rescue mission. It was a big error. Let me say a bit about Fisher, if you don't mind.
Please.
That would be great, yeah.
I met him and I knew him. I dined with him twice. He was an absolutely brilliant dinner table conversationalist. He could talk about anything and everything in an amusing and interesting way. He obviously had a mind which was 20 levels higher than just about that of anybody else. He was totally amazing. He was also an utterly total, you better not use the word. Did I tell you the example of that? So I went to a seminar which he gave, and he was going along in his talk, and he came to a rather dubious point. And I was a graduate student, master's graduate student at the time, and another graduate student put up his hand and said, "Professor Fisher, could you just go back over that point and just go through it again?" He was really saying what you said was very obscure and you should clarify. What Fisher said was, if you are too stupid to understand what I'm saying, you shouldn't be here. And just kept on going. That is what he was like. He was brutal. That's totally unacceptable behavior. Totally unacceptable. I also got to know Wright pretty well on my visits to Madison. He was a real gentleman, a real nice guy, but he didn't sparkle anyway like Fisher did. He was muted, very quiet, very I hope I don't insult people by saying this, very Midwestern, very down to earth and practical, not trying to sparkle all over the place. So I knew them both reasonably well.
Did you ever meet Haldane?
No, he's the one person I never met. I'm pretty glad about that. I don't think I could have got on with him.
Okay, yeah. So thank you for your reflections on Wright and on Fisher and on Moran, because those are people that we can't meet, and so I really appreciate the stories.
Another way of saying what you're saying, Sally, is I'm an old fuddy-duddy and only think of the past.
Well, these stories connect us to the past, and so they're really golden, and I appreciate that.
Yeah.
Thank you so much, Warren, for joining us today.
That was a pleasure.
Thank you.